for those who saw the previous video, this video is also very helpful to understand all the enzymes and all the things. So it's going to be very fundamental. It's going to take some time, but we know that enzymes are proteins, okay? And we know that proteins are synthesized from mRNA, mRNA, tRNA, and rRNA. And the central dogma says DNA becomes proteins. These are the things we know. And we know that alternative splicing can produce a variety of protein products. For example, different types of immunoglobulin, tropomyosin variants, dopamine receptors, insert D1 or D2, or muscarinic M1, M2, M3, M4, M5. These could all have alternate splicing. And so quickly, we know that these things in molecular biology form a protein. After the protein is formed, it undergoes something called post-transcriptional modification. And this post-transcriptional, translational, post-transcriptional, and then post-translational modification could be something like trimming, such as conversion of trypsinogen to trypsin, as a gastric trimming, or the entire body, the entire biochemistry is based on this post-transcriptional modification, which enzymes do covalent alterations. Now, covalent alterations are where the entire biochemistry gets unlocked. And covalent alterations such as phosphorylation, phosphate group is the backbone of energy. ATP, ADP, AMP, adenosine, carbamoyl phosphate, heme, all those things. Glycosylation, adding of sugar, hydroxylation, methylation, acetylation, ubiquination. These are the covalent alterations that can happen. There's something called a chaperone protein, which pH, hypoxia, and all of that causes, like an RBC in sickle cells against denature. So, if you understand this post-transcriptional modifications, we'll jump into enzymology. So here you understand the enzyme terminology. Okay, so these are the modifications that happen to proteins. Okay, so an enzyme's name describes its function. So anything with kinase in it. So any enzyme with kinase transfers a phosphate group from ATP. So, for example, glucokinase is the one that transfers phosphate group from ATP. And glucokinase causes, when there is energy, transfer the phosphate group to glucose. That means you will form glycogen because there is energy. If there is no energy, then you will have something like phosphatase, glucose 6 phosphatase. That is the enzyme which is deficient in McArdle's disease. Therefore, you will have cellular death. These and phosphorylase. What is phosphorylase? Adds an inorganic phosphate without using ATP. So this is not that critical, but phosphatase as an enzyme removes the phosphate group, glucose 6 phosphatase. When will it remove it? When there is no energy. Kinase is when there is extra energy. So kinase would cause storage. Phosphatase would cause breakdown. Okay, so that's the first covalent modification you need to understand. Something to do with the phosphate group. The next covalent modification is what happens with HOH. So dehydrogenase hydroxylase hydroxyl group adds a hydroxyl group, tyrosine hydroxylase, scurvy, vitamin C. Carboxylase transfers carbon dioxide group with the help of biotin bias, carboxylase. Mutase synthase joins two products together. So with this terminology, now we break down biochemistry and post-transcriptional modifications. And now we have terminology. And now you see how it gets fun. So, rate determining steps of glycolysis. What's glycolysis? Breakdown of glycogen. Phosphofructokinase. What's a kinase? Catalyzes the transfer group from a high energy. So to start breaking down sugar, first we need old energy. So ATP, AMP. So this is driven by AMP, phosphofructokinase. ATP inhibits this because we have energy. Okay. Now the gluconeogenesis is to make glucose. Correct? When there isn't any glucose. See the enzyme phosphatase. What does that mean? Fructose 1,6,5 phosphatase. What does this mean? So what do you think AMP will do? AMP will see you have either ATP or AMP, correct? So ATP, do you think ATP will promote gluconeogenesis? No. ATP will inhibit it. AMP also negatively inhibits it. TCA cycle. Isocitrate dehydrogenase. ADP, ATP, NADH. So ATP will prevent TCA cycle from happening. ADP will promote it. Glycogenesis, glycogen synthase. Glucose 6-phosphate enzyme promotes glycogen. Insulin promotes glycogen. Cortisol promotes glycogen. Epinephrine prevents glycogen. One glucagon wants glucose. Epinephrine promotes glycogenolysis. Genesis genolysis. HMP shunt. NADP promotes it. NADPH prevents it. So the enzymes are like always bidirectional and they have a bidirectional impact on the substrate. And they decide what to do based on which substrate and regulator is available. ATP promotes pyrimidine synthesis. Urea cycle, Na-glutamate, carbamyl phosphate, acetyl-CoA carboxylate. So basically, what you need to understand is this opens the world of this beautiful thing, which all makes sense now that you know the terminologies. So what's a kinase? There's so many kinases. What's a kinase? Look at number one. Galactose becomes galactose 1-phosphate. Galactose 1-phosphate becomes glucose 1-phosphate. And then, so kinase is the one that adds a phosphate group from galactose. So think of a phosphate group as a tag here. When galactose gets tagged with the phosphate group, it's blocked for getting converted to glucose, and that happens through galactose 1 phosphate uridyl transferase, and then uh, that becomes UDP glucose, and then that becomes glycogen. Okay, so glucose 1 phosphate to become glycogen has to become UDP glucose, and then it becomes glycogen. But now look at this. Now you have glucose. Correct? Glucose came from food. Now what has to happen to glucose? Glucose has to become acetyl-CoA. Look at the terminology. Glucose 6-phosphatase. What do you think glucose 6-phosphatase will do? Number 4. That is ulta. That is going from glucose 6-phosphate to glucose, not this way around. But when there is glucokinase with ATP, when ATP is there, it takes ATP and forces it down because it's a difficult process, but you need that ATP here. So that's glucokinase. Glucose 6-phosphate dehydrogenase, which forms ribulose, and then this goes for purines and pyrimidines. Phosphatase. So breaking this fructose from glyceraldehyde DAP. 3-1 PEP, pyruvate dehydrogenase complex, acetyl-CoA, and that's how you read enzyme reactions. Now, see this. This where it comes very interesting. What are vitamins? Vitamins are nothing but the coenzymes. They act along with the enzymes and they all have a particular function. B12 has one function to transfer a methyl group. What's the function of B12? It helps methylation. What's the function of methylation in DNA? That's a whole different thing. But what's the function of methylation of homocysteine? It becomes methionine. Methionine becomes SAM. SAM then provides methylation indirectly, and then SAH becomes adenosine. What does B6 do now? Say B6. What's the function? Transamination. Transfers amino group, NH3 group. So what do you think homocysteine within a transamination reaction would give it to cysteine? Okay, so you need B6 for that. What does vitamin C do? Hydroxylation. And it keeps everything reduced. That is not the main function of vitamin C. It's acidic, so therefore it keeps it reduced to Fe2+. But hydroxylation is the main function. So if you need to hydroxylate an enzyme, use vitamin C. If you need to methylate an enzyme, use vitamin B12. If you need to do a transamination reaction, use vitamin B6. If you need to buy or add multiple carbons, use B7 called biotin. If you need to convert tetrahydrofolate for one carbon transfer reaction, tetrahydrofolate, use B9. Folate. B5 is a coenzyme for CoA. Acetyl-CoA. B3 is oxidation reduction, just an electron. B2 is also an electron. B1 is a cofactor for pyrophosphates. And B1 deficiency causes, thiamine deficiency causes wernicke encephalopathy, Korsakoff syndrome, tri and beriberi. Because these are all neurological conditions where phosphating pyruvate dehydrogenase transketolase alpha ketodehydrogenase. This is all the complex of P1. It is called the pyruvate dehydrogenase complex, which is very important. It links the glycolysis to TCA. Also, it is what makes transketolase possible. Even a branched chain keto. So that's water soluble. What about fat soluble? Fat soluble vitamins. Vitamin A is an antioxidant. Vitamin D, calcium. Vitamin K, clotting. Vitamin E, oxidation reduction. Zinc. So basically, that's how you need to break biochemistry down. This can go on forever. But now, when you look at these reactions, it looks better. NAD to NADH. What's this? This is vitamin D, B3. You already know that. In B6, in pyruvate dehydrogenase complex, B1. So you can answer more questions. We'll discuss more again.